To a new module in this lecture series. Today we are going to look into the second module of this series, which is in which we will be exploring more about C3 leaf physiology. So in the last class, I had given a brief overview of what are the different photosynthetic pathways we can see in plants. Basically, the C3, C4, and the CAM photosynthesis. Now, all of these lectures are going to explore more about the C3 leaf C3 photosynthesis and what are the equations and models that can be used to obtain the C3 leaf photosynthesis and the various processes related to that. So, like I had explained earlier, all the light dependent reaction and Calvin cycle was related to C3 leaf photosynthesis. The light dependent reaction is the same in most of it. The electron transport chain, which is basically the light dependent reaction, is similar in C3, C4, and CAM. But when it comes to the Calvin cycle in C3, it is happening in the stroma part, and there is only one cycle, this Calvin cycle, with also photorespiration occurring in C3, where RUBP is combining with carbon dioxide with the help of Rubisco enzyme and reducing into G3P, which is later on used for sugar, for manufacturing sugar and regeneration of RUBP. So this is the light dependent reaction, and this is the Calvin-Benson cycle that is occurring in C3 photosynthesis. Again, photorespiration is also a process that occurs, which I had explained in the previous lecture, where instead of carbon dioxide, RLBP reacts with oxygen to form PGA, phosphoglycolate and PGA, and which gets converted into PGA later on through this cycle, where part of it happens in chloroplast. Peroxisome glycolate gets converted into glycine, and then mitochondria, glycine gets converted to serine, during the course of which a carbon dioxide molecule is utilized and removed from this Calvin cycle. So this is considered a wasteful cycle, considering the removal of carbon dioxide from the Calvin cycle and also the stable compound that is formed later on is not enough for RUBP regeneration, neither is it enough for carbohydrate carbohydrate manufacture. Now, another important process that is related to photosynthesis is the dark respiration. Important process, dark respiration, which is also called as growth and maintenance respiration, occurs in plants, and unlike photorespiration, this is a very important respiratory process happening in plants to maintain the energy and promote growth for the plants. So, dark respiration is the process in which plant cells are breaking down the glucose that is obtained during the photosynthetic process to produce energy in the absence of light. So, this respiratory process, unlike photorespiration, doesn't need light, and it is a very important process for the plant to grow and maintain its energy. And this dark respiration occurs in the mitochondria of plant cells. Now, while photosynthesis is using light energy to convert carbon dioxide and water into glucose and oxygen, dark respiration is using oxygen. That is, it takes up oxygen and then uses the glucose that is produced during photosynthesis to produce carbon dioxide, water, and ATP. Now, unlike photosynthesis, which requires light, this process does not require light, and since it's a respiratory process, it is using up oxygen. So, irrespective of time, it is always happening, day, night, as long as the plant is alive and it is making use of the energy for various purposes, it is always happening. Now, the processes that are involved in the growth respiration or dark respiration includes glycolysis, the citric acid cycle, and the electron transport chain, all of which is taking place in the mitochondria. So, like photosynthesis and photorespiration, growth and maintenance respiration also happens in the form of a bunch of cycles. First process is glycolysis, and then it enters the citric acid cycle, and there is also an electron transport chain that is electron transport occurring during the growth and maintenance respiration. Dark respiration again is not requiring light, but it is indirectly related to photosynthesis because it is the glucose that is produced during photosynthesis that is used up during respiration. So it is like the fuel for respiration, and it releases the energy that will be used for growth, that will be used for repair of the plant cells and other cellular activities. So even when it's dark and plants aren't photosynthesizing, it still continues to use the glucose that is produced from produced during photosynthesis via this dark respiration. So in C3 plants, mitochondria can be seen in many places, primarily found in mesophyll cells that we talked about, palisade and spongy mesophyll cell. It is also seen in bundle sheath cells. In C3, if you had seen, the bundle sheath cells were not very prominent, prominent as you could see in the C4 photosynthesis. So mitochondria is present in this bundle sheath cells also. Guard cells are the cells that surround the stomata. They also have mitochondria. Root cells have mitochondria, and meristematic cells. These are actively dividing cells, which you can find in the tip of the shoots and tip of the root. So this is what I said, and these are just pictorial representations of that. So if you look at a plant cell, you can see mitochondria here. If you look at a roots, in the end of the roots, tip of the root, you will find mitochondria and the cells at the tip of the root. Now, if you see here, apical meristem is nothing but actively dividing cells. So they keep dividing so that this stem will grow in height. So in these cells also you can find mitochondria. Same in the condition of roots also. They keep developing so that the roots grow vertically or in the vertical direction downwards, and in those places also you will be able to find mitochondria apart from plant cell, guard cells, etc. So this is basically the cycle that is happening during dark respiration. The dark respiration or the growth and maintenance respiration occurs it's a combination of three cycles or three processes, namely glycolysis, Krebs cycle. Krebs cycle is also called as the citric acid cycle, and electron transport chain. So let us see how this occurs. So glucose, which is generated from the process of photosynthesis, will get converted into pyruvate during the process of glycolysis. So glycolysis happens, glucose gets converted into pyruvate. Pyruvate then gets converted into acetyl CoA. Now these three processes are happening in the cytoplasm of the cell. That is if this is the mitochondria, it is happening outside the mitochondria in the cytoplasm. So glucose get converted to pyruvate, and pyruvate gets converted to acetyl CoA and enters the Krebs cycle, or it is also called the citric acid cycle. So in the Krebs cycle, what happens is acetyl CoA gets converted into carbon dioxide, ATP, and NADH and FADH2. So if you see the cycle here, acetyl CoA is getting converted to citric acid, then it is getting converted to isocitrate, ketoglutarate, succinyl CoA, succinate, fumarate, malate, and back to oxaloacetate. So during this course of reaction, you will see that there is something called NADH formed, not NADP. That was in the light dependent reaction. NADH formed, and you will also see NADH formed during this portion of the cycle. You will also see ATP is synthesized during this part of the cycle, and you will see another molecule called FADH2 that is produced during this part of the cycle, and again NADH. So if you see here, NADH is formed, ATP is formed, FADH2 is formed, and you will also see that carbon dioxide is being released during this cycle. So the output of this citric acid cycle is carbon dioxide, NADH and FADH2, and a bunch of ATP molecules. Now even during glycolysis ATP is produced, take note of that, and here in Krebs cycle also ATP is produced. Apart from that, carbon dioxide is released, and NADH and FADH2 that is produced during the Krebs cycle are sources of high energy electrons, because they are getting they are accepting electrons over, they are releasing H plus and getting reduced. So NADH and FADH2 are high sources of electrons, and when these high sources of electrons provide their electrons to this electron transport chain, this electron transport chain that is occurring in the membrane of the mitochondria, similar to the membrane of the thylakoid where all the electron transport chain is occurring. This membrane is also having a bunch of integral protein complexes, and this electron will get transferred from one protein to another, and during this cycle an energy gradient is formed. So this energy gradient is helping in creating a proton gradient, because as this electrons are getting moved from one place to another, H plus ions of this H, it forms a proton gradient for the H plus ions, which will in turn help ATP synthase again present in this mitochondrial membrane to generate a lot of ATP molecules. So during the electron transport chain, electron transport occurs, which is provided by this NADH and FADH2, and proton grade, as a result of the proton gradient, ATP synthase produces a lot of ATP molecule. The final acceptors of the electrons are oxygen, and oxygen, and then finally water molecules are formed as an output of this electron transport chain. So when you see this entire cycle, we don't have to go in too much detail of this Krebs cycle, glycolysis, and electron transport chain in dark respiration, because our models are more focused on the photosynthetic part. Dark respiration is also used as a parameter, but not as extensively as photosynthetic parameters are used. So we are learning about dark respiration and what happens inside it, although we are not extensively modeling this entire process within BioPro. So that is why I am not going too much in detail into this process. So totally, the whole this whole process is generating a lot of energy for the plants for growth and repair. So again, I have just mentioned the major steps in here. Glycolysis occurring where pyruvate is formed. Then during this reaction, there is ATP, NADH, and pyruvate molecules formed. Citric acid cycle or the Krebs cycle occurs when pyruvate that is converted into acetyl CoA enters the Krebs cycle, and they produce carbon dioxide, NADH and FADH2 and ATP. Then NADH2 and FADH2 donate high energy electrons to the electron transport chain. They move through the protein complexes, pumping proteins across the membrane, creating a protein gradient, which drives the ATP synthase to produce a bulk of ATP, and oxygen acts as a final electron acceptor, forming H2O. So right now we learned what is light dependent reaction, Calvin cycle, photorespiration and dark respiration processes. Now the next and the very important step when it comes to modeling is to understand what equations are used to represent these processes in a plant model. So these, if you see, what all we studied are the theoretical, are the processes that are happening, and what kind of reactions and what kind of physical processes are happening when photosynthesis and photorespiration and dark respiration occurs. Now, how do we represent all of these processes mathematically and into a computer model is what is where a lot of equations and equations developed by a lot of prominent scientists uh, come to our assistance. So one of that is this model or an equation called the Farquhar, von Kammerer and Berry model. So this is the first set of equations we will be studying that is used extensively in BioCro and various other crop growth models that represent photosynthesis. Photosynthesis for, and particularly to be used at leaf scale. Now, Farquhar von Kammerer and Beatty model is a biochemical model. So, when I say a model is nothing but a set of equations that are used in representing a process. So, don't get confused with the BioCro. BioCro is a package that comprises a lot of equations like this, which will help in simulating or representing the plant growth in a computer. And one of the set of equation is this Farquhar-Von Kammerer-Berry model, FvCB model. It is a biochemical model that is describing the rate of carbon dioxide assimilation in tree plants, and it is linking the leaf gas exchange to enzymatic processes within the chloroplast. It was first introduced by G.D. Farquhar, S. Von Kammerer, and J. Berry in 1980, and it is representing the rate of net photosynthesis as being limited by three key biochemical processes. So, this model is giving us the rate of net photosynthesis, and this net photosynthesis, the main factors that are limiting this net photosynthesis are three biochemical processes, namely the Rubisco limited phase, RUBP regeneration limited phase, and triose phosphate utilization limited phase. Rubisco limited phase or carboxylation limited phase, that is represented by the letter A with the subscript C in the FvCB set of equations, is when the rate of carbon dioxide assimilation. So when we say assimilation, we mean the rate of net photosynthesis. What is the net rate of carbon dioxide that is accumulated within the plant cells for photosynthesis? When the carbon dioxide assimilation is constrained by the activity of Rubisco. So we know Rubisco is a very important enzyme in the Calvin cycle, and when the carbon dioxide assimilation is constrained, that is Rubisco acts as a limiting factor when carbon dioxide assimilation occurs, then it is called the Rubisco limited phase. Similarly, when assimilation is limited by the regeneration of RUBP via the Calvin cycle. So during Calvin cycle, we have also learned about how RUBP is regenerated, and when this RUBP regeneration is limiting, that is because of RUBP regeneration at a very high or very low levels, if the carbon dioxide assimilation rate is affected, then that phase is called the RUBP regeneration limited phase. Similarly, triose phosphate utilization limited phase is nothing but a phase where when the carbon dioxide assimilation is limited or is constrained by the capacity of exporting triose phosphate. To export and use this triose phosphate, then it is called a triose phosphate utilization limited phase. Now, the conditions during which all of these limitations occurs is when there is low carbon dioxide concentration and when RUBP is sufficiently available. What happens is RUBP will not have enough carbon dioxide to for the reaction to occur. So, Rubisco is limiting it because the carbon dioxide concentration is less, and in place of that, probably photorespiration will occur because of the low concentration of carbon dioxide molecules. So in this case, we say the Rubisco is limiting this assimilation rate. Now in the case of RUBP regeneration, when there is moderate to high carbon dioxide concentration and when Rubisco is saturated with carbon dioxide and the limitation shifts to ATP and NADPH production from electron transport, then RUBP regeneration limited, we call that carbon dioxide is limited by RUBP regeneration. That is, when there are moderate to high concentrations of carbon dioxide, Rubisco enzyme is saturated with carbon dioxide. It is not able to take in more of carbon dioxide for attaching RUBP with carbon dioxide. So in under such cases, the limitation is occurring because of the less amount of ATP and NADPH production from electron transport. So that is why it is called RUBP regeneration limited phase, and it is also called the electron transport limited phase. So this is the connection. When Rubisco is saturated with carbon dioxide and limitation is shifting to the amount of ATP and NADPH production during electron transport, which is going to impact the carbon dioxide assimilation rate. So that is why this is an RUBP regeneration of limitation due to regeneration of RUBP or electron transport is often interchangeably used. Now the next limiting phase, which is the triose phosphate utilization limited phase or TPU limited phase, is more common at very high concentrations of carbon dioxide, where the starch and starch content synthesis becomes limiting. So when high concentration of carbon dioxide occurs, there is a tendency for the starch synthesis to become limiting because triose phosphate is not sufficiently utilized. So under such conditions, the TPU becomes a limiting factor. So if you had noticed in the Calvin cycle, we had seen this is the carbon fixation, carboxylation, reduction, regeneration. These are the three processes that are happening during the Calvin cycle. So this first process, which is carboxylation, is one limiting factor for carbon dioxide assimilation. The second limitation we saw is the regeneration of RUBP, that is also limiting the net rate of carbon dioxide assimilation. And the third is triose phosphate utilization. What triose phosphate is nothing but the G3P molecule that is formed as a reduction of this PGA molecules. So uh, G3P is called triose phosphate, and this triose phosphate has to be properly utilized for glucose to be manufactured. If it is not properly utilized, again there is an accumulation of which will again limit the carbon dioxide process. So most of this limitation is occurring in the as a result of processes happening in the Calvin cycle. Again, in the regeneration of RUBP, this high concentration of carbon dioxide is saturating Rubisco, as a result of which the limitation is getting limitation is will get transferred to the amount of ATP and NADPH that is produced during the electron transport chain. So that is one that is associated with the light dependent reaction, and these two processes are associated with the Calvin cycle. So this is the main equation of the FvCB model. The FvCB model's main equation is that A net will be whichever is the minimum of these three processes, that is the carboxylation limited, the RUBP regeneration limited, or triose phosphate utilization limited photosynthetic rate, whichever is the least of these three limited photosynthetic rates, that is taken, and that value is subtracted from the dark respiration or growth or maintenance respiration, and then the whatever carbon dioxide assimilation you get will be the net carbon dioxide assimilation produced at that particular point or particular leaf or particular time period or whatever. So here, like I had earlier mentioned, AC is the Rubisco limited photosynthesis, which is also called carboxylation limited. AJ is the RUBP regeneration limited photosynthesis, which is also called electron transport limited. And AP is the TPU limited, triose phosphate utilization limited photosynthesis. ANet is the net assimilation rate. RD is the dark respiration rate, which is also called as the mitochondrial respiration or growth and maintenance respiration. So this is the main equation of FvCB model. Now our next part is to see how AC, AJ, AP and RD can be obtained. So how do we obtain the values of AC, AJ, AP and RD? So first we look into the Rubisco limited photosynthesis, carboxylation limited photosynthetic rate. So in order to obtain AC, we need all of these parameters to calculate the value of AC. So let us look into this equation and what are these parameters. So Vc max into Ci minus tau star, or we can for E say it is G star, divided by Ci plus Kc into 1 plus O by K naught. So what are each of these terms is what we look into now. So Vc max is nothing but the maximum carboxylation rate of Rubisco. That is, when we said that this Rubisco limited photosynthesis occurs, this value Vc max is representing the maximum rate of carboxylation. Carboxylation is nothing but the reaction of RUBP with carbon dioxide. So what is the maximum rate at which carboxylation is happening as a result of Rubisco is what this parameter called Vc max depicts. Ci is nothing but what is the intercellular concentration of carbon dioxide, that is within the plant cell. We had looked into a leaf cross section and you had seen that carbon dioxide and oxygen is circulating within the spongy mesophyll. So what is the concentration of carbon dioxide within a leaf is what Ci indicates. Now next is G star or tau star, which is called the carbon dioxide condensation point in the absence of mitochondrial respiration. So this is nothing but the theoretical, it's a theoretical value. It is not an actual value that that you can measure and obtain. It is a theoretical value of carbon dioxide concentration within the leaf when the net carbon dioxide assimilation rate is zero. That is when the carbon dioxide uptake during carboxylation is equal to the carbon dioxide released by photorespiration, then that point, that 0 point at which the that is when the uptake is equal to the release of carbon dioxide due to photosynthesis, then that concentration of carbon dioxide within the leaf is called the carbon dioxide compensation point. So I think I can explain more when we come into the later slides of carbon light response, I mean photosynthetic response curve. So I will uh, stop with this explanation of carbon dioxide compensation point, which is nothing but the theoretical concentration of carbon dioxide inside the leaf when the net uptake of carbon dioxide is equal to the net release of carbon dioxide during photorespiration. Now the next is this Kc value. It is the Michaelis-Menten constant for carbon dioxide, which also I will be explaining. O is the oxygen concentration inside the leaf. K naught is the Michaelis-Menten constant for oxygen. So what is the details of these parameters will be explained to in later slides, because we need to understand these parameters well in order to when we are using the model. We can use the model without knowing all of this, but if we need to do an extensive analysis seeing which parameters is affecting the process, then we have to completely understand all of these parameters. So in Rubisco limited photosynthetic rate, this Vc max is a very important parameter because it is representing the maximum rate at which carboxylation occurs. Ci is the intercellular carbon dioxide concentration. G star is the carbon dioxide concentration during which the net uptake of photosynthetic rate is equal to the net release of carbon dioxide when photorespiration occurs. It is called the carbon dioxide compensation point. Kc and K naught are the Michaelis-Menten constants for carbon dioxide and oxygen, and O is the oxygen concentration inside the leaf. Now this is how the Rubisco limited photosynthetic rate is depicted. The next is the RUBP regeneration limited photosynthetic rate. So similar to the AC, we see it is represented by J, AJ, which is equal to J into Ci minus tau star by EC minus into Ci plus 2 E naught minus into tau star. So if you see here, you see Ci minus tau star is a common factor. Here also Ci minus tau star is a common factor. So same like in AC, this Ci means intercellular carbon dioxide concentration, tau star is the carbon dioxide compensation point, J is the rate of electron transport rate. That is, what is the rate at which electron transport is occurring is what is J. And E C minus is how many electrons, the amount of electrons that are used for carboxylation and amount of electrons that are used for oxygenation. That is um, when photorespiration is occurring, RUBP is combining with oxygen. So how many electrons are used for this oxygenation process and how many electrons are used for this carboxylation process are what this E terms represent. Basically, amount of electrons used during carboxylation and oxygenation. So this is how RUBP regeneration limited photosynthetic rate is depicted. Now we come to the TPU limited photosynthetic rate. In the TPU limited photosynthetic rate, the equation for estimating the assimilation rate limited by triose phosphate utilization is 3 into VTPU. VTPU is nothing but the maximum rate at which triose phosphate is being utilized. Now, I had earlier said what triose phosphate is. Triose phosphate is nothing but this G3P. G3P is also called triose phosphate. Now, this triose phosphate, when generated, is used in the manufacture of carbohydrates, is what we learned during Calvin cycle. So, once this triose phosphate is used, what is the rate at which this triose phosphate is utilized for the manufacture of glucose is what is given here. Now, we know Calvin cycle is happening in the stroma of the chloroplast, and from there, the triose phosphate that is generated from the Calvin cycle will move into the cytosol. For this, whatever cycle is responsible for making use of generation of carbohydrates, and during that process, when this triose phosphate is sent into the cytosol or cytoplasm from the stroma, the cytosol is providing in return a inorganic phosphate molecule back into the stroma. Now, if you see here, this inorganic phosphate is what will react or what will attach, what will combine with the ADP to form ATP during the light dependent reaction. We have seen that proton gradient is formed, ATP synthase is driven by this proton gradient, and then ADP along with an inorganic phosphate will form ATP. So where is this inorganic phosphate coming? It is coming from cytosol when this triose phosphate during Calvin cycle is pushed into the cytosol for the manufacture of starch. So this is the maximum rate at which the triose phosphate is utilized is another limiting factor, and the photosynthetic rate corresponding to this maximum rate of triose phosphate utilization is depicted by AP and is called TPU limited photosynthesis. So now we have the FvCB model which takes the minimum of AC, AG and AP. AC is this given by this equation, AG is given by this equation, AP is given by this equation. So when you estimate, when you put in all these parameters and when you estimate the net photosynthetic, the corresponding photosynthetic rate, we have to choose the minimum of these three values and then subtract it from the dark respiration rate to get the net photosynthetic rate. So this is what the FvCB model is all about.